Hello, my name is Mudassir Muhammad. I'm a technical support specialist here at Sigma Life Science. Today I'm going to introduce you to chemistries for challenging qPCR assays. There are a number of alternative probe chemistries to overcome assay design problems, optimizing the assay by improving sensitivity and discrimination. There are a huge number of chemistry that is used for detection. However, each have their own advantages. For some assays with low copy number, the use of alternative detection chemistry can improve the quality of the assay compared to using a Techman probe, as they have additional properties to improve their sensitivity to a target. Each chemistry has its own advantages. Molecular beacons are ideal for in vivo assays and can improve specificity around a SNP. Scorpion probes can improve sensitivity if you have a low target yield or a very short region. Locked nucleic acids or LNAs can improve sensitivity. These are good in AT-rich sites. If you have a short region, you can design a shorter LNA probe which can improve sensitivity, maintain the primer's probe design rules. We'll go through each of these alternative detection chemistries as they differ from standard dual label fluorescent probes. All are specific probe-based chemistries, and fluorescence is based on threat. This slide shows the molecular beacon. Beacons are developed as a method to detect SNPs, a single base changes in a template, and are also good for in vivo study as the formation of a hairpin structure protects the probe to a certain extent from nucleus activity. Living cells have an array of functional enzymes with nucleus activities that would degrade the probe. The two complementary arm sequences 4 to 6 mer, which are not part of the target sequence, form a hairpin loop structure. The hairpin brings the quencher in close proximity to the fluorophore, effectively quenching it. A probe sequence is longer than the arm sequence, therefore, in the presence of the probe sequence, the kinetics of the target will be greater than the arms, allowing it to bind to the target sequence. These slides demonstrate how molecular beacons work. There is no fluorescence in solution, as the hairpin quenches any fluorescent signal. Like Techman probe, molecular beacons binds to the target before the primers. Probe is in the center of the target region and not close to the primer to encourage displacement rather than hydrolysis. During extension, the Taq polymerase displaces the beacon and it goes back into solution. Therefore, we measure fluorescence during the annealing phase of the reaction. When using molecular beacons probe for qPCR, it is often recommended a 3-step qPCR cycle with separate annealing and extension steps. This is the PCR cycle used for standard endpoint PCR, such as denaturation at 95 degrees, primer annealing at 65, and extension of the Taq at 72 to 74 degrees. By raising the temperature to 72 degrees, Taq is at optimal kinetic temperature to function, which is enzymatically favorable and moves along strand more quickly to encourage displacement. This is why amplicon is also situated further away from the primer. This slide shows the advantages and disadvantages of using molecular beacons. Molecular beacons probe can be more specific than dual label fluorescence probe, as molecular beacons are less stable during annealing. Hence they are good for SNP detection, where the TM is approximately 65 to 70 degrees. It is harder to design the probes, as they have to be designed over a SNP region. SNP region may be poor quality with high homology of folding. Hence molecular beacons are more reliable than Techman probe over a SNP. We have talked about dual label fluorescence probes and molecular beacons. The third type of probes are called Scorpion probes. These probes are shorter and have greater sensitivity and design flexibility. Scorpions are very specialized probes and are often used when Tachman and molecular beacon cannot be used. Example, poor binding regions or very low copy number, as these are one of the most sensitive detection chemistries available. Scorpion probes resemble the stemp loop structure of molecular beacon. With dual label fluorescent probes and molecular beacon, the probe is separate to the primers. However, with Scorpion probe, the probe is bound to one of the primers. A diagram shows a Scorpion in the presence of the probe is extended from the 5' end of the primer away from the 3' end, where extension occurs. A blocker is inserted between the primer and probe. Therefore, during PCR, the Tachman will not extend past the primer and the probe. Scorpion uniprobes consist of a single stranded sequence held in a hairpin loop conformation, approximately 20 to 25 nucleotides, by complementary stem sequence, which are approximately 4 to 6 nucleotides on both ends of the probe. The probe contains a 5' end reporter dye and an internal quencher dye directly linked to the 5' end of the PCR primer via a blocker. The close proximity of the reporter dye to the quencher dye causes the quenching of the reporter natural fluorescence, similar to the molecular beacons. The Scorpion probes are designed with a distance of more or less 50 base pairs from primers. The Tachman and molecular beacon probes can be designed on both sense and antisense strands, as can be Scorpions as well. However, if the probe is attached to the sense primer, then it must be an antisense probe that will target the region of interest on the same strand. This and the following slide show how the Scorpion works. The primer probe binds to the target and undergoes normal extension via the Tag polymerase, amplifying the target. The same occurs on the second strand with the standard primer. Denaturing the double-stranded DNA causes the two strands to separate. As the strands separate, the probe opens up and twists back to the complementary region on its own strand. This effectively separates the fluorophore from the quencher, allowing it to fluoresce. This is because it is more kinetically favorable than binding to the hairpin conformation. As the tail of the Scorpion and PCR product are now part of the same strand of DNA, the detection interaction is intramolecular and so a much more rapid reaction than comparable probe-based detection systems. The target sequence is typically chosen to be between 5 and 50 bases of the 3' end of the Scorpion primer. Scorpions are very sensitive and provide a strong and rapid signal under fast cycling conditions compared to dual label fluorescent probes and molecular beacons. Here are some advantages and also few advantages of using Scorpion probes. Scorpions are more sensitive than other probes, as the probe is attached to the primer, guaranteeing that after extension, the probe will bind to the target. Tachman and molecular beacons rely on the probes in the solution to find their way to the target and binding. Hence, they are less sensitive compared to Scorpion probe. The Scorpions allow shorter probes to be designed and increase the sensitivity and design flexibility. Disadvantages are these are expensive and often require complex synthesis and design criteria. This slide shows you locked nucleic acids or LNA. Up until now, I've discussed the different structure of probe and how this can be used for different types of analysis. However, another type of probe is an LNA probe. The LNA contains a methylene bridge. The bridge restricts flexibility of the ribofuranose ring and locks the structure into a rigid C3 endo conformation. Hence, the backbone of the oligo is altered. LNAs are synthesized in the same manner as DNA. LNA can confer enhanced hybridization and stability compared to DNA. LNAs are also compatible with RT-PCR platforms and endpoint analytical detection instruments, and soluble in standard buffers and solutions. They are very good at recognizing inaccessible SNP to improve discrimination of relatively stable mismatches. There are advantages of incorporating LNA into other probes as well, such as dual label fluorescence probe, molecular beacons, and light cyclers. Because a shorter sequence is required when incorporating the LNAs into the probes, it allows greater flexibility in the design. When designing probes for qPCR, they need to have a TM of 10 degree higher, so the primer anneal first. If amplicon target region you are working with is short, you can raise the TM of the probe by inserting LNA, as LNA increases the TM without the requirement to add extra nucleotides into the probes, which can potentially help target regions of poor binding, such as AT or GC-rich regions. However, in the next two slides, I'm going to show you how the incorporation of LNA bases into your probe design can help improve the sensitivity of assays for mismatch detection and improve the specificity of the probe. This chart shows the sensitivity or discrimination of a SNP detection with and without the incorporation of LNA into probes. The probes here are 16 mers with 3 LNAs. As you can see on the right hand side, there is a huge discrimination of the mutant and the wild type when LNAs are used as compared to when there are no LNAs within the probe. This slide shows you how the incorporation of LNA bases into your probe design can help improve the sensitivity of the assays for mismatch detection and improve the specificity of the probe. The gel image shows the DNA versus LNA mismatch extension analysis using the PUC19 plasmid. The panel A on the left shows DNA primers showing mismatch extension in 2 to 3 possible mismatches. The panel B on the right shows PCR primers with a 3' LNA base, eliminating the mismatch extensions seen with the DNA primers on the left. There are certain advantages and disadvantages of using LNA as well. The advantages are when LNAs are used, one can increase the sensitivity and specificity of the assay. One can also improve the mismatch discrimination, and I have briefly shown you in the previous slide. Incorporation of LNA allows shorter probe to be designed, hence it increases design flexibility. The LNAs are also good for in vivo studies, as the backbone is protected to a certain extent from endonucleases within the cells. Disadvantages is these are expensive and also of secondary structures. Here's the table of detection chemistries. Choosing the correct chemistry for qPCR assays is important. This table lists the detection chemistry available, and while they can be used in many qPCR applications, more suitable for some depending on what you are looking for. In summary, I'll briefly describe the selection of appropriate qPCR chemistries. Apart from dual label probes, there are other chemistries, such as molecular beacons we have briefly gone through, and also complex Scorpion probes. The incorporation of locked nucleic acids have been described, and the incorporation of these into probes can increase specificity, increase the TM of the probes, and in vivo studies these can be used as the LNA incorporation can protect the probe from endonuclease activities within the cells. If you would like some assistance with the design, you can contact us via www.sigma.com/designmyprobe. Once you enter this site, you'll be taken to our page called OligoArchitect Primer and Probe Design Solutions. You can either click on OligoArchitect Online or OligoArchitect Consultative Design. OligoArchitect Online allows you to perform an online design by yourself. An OligoArchitect Consultative Design, alternatively, allows an expert to create a design for you. As we provide this as a free design service, and the feedback we received of the essays that we have designed have been very positive, please feel free to submit your designs using this website. Apart from essays designs, we also provide essay optimization, troubleshooting, and online seminars. Thank you very much for listening. Bye-bye.